morning, students. So in our previous class, we come to know about the actual meaning of a heredity, and then we come to know about the variation and the proper definition of a genetics, and whatever the terms we are going to see in the complete chapter, like inheritance and the variation. Okay. So today we are going to deal with the next concept, that is about Gregor John Mendel. Okay. So before talking about that person, I will tell that first, please subscribe to our channel SLT PU College of Science, Gurumatkal. Okay. And if you get any doubt after the completion of this concept, if you get any doubt, so please ask the questions in a comment box. Okay. So now we are going to discuss about the Gregor John Mendel. Okay. So the complete genetic concept has been developed by the Gregor John Mendel. So that is the reason we call that person as a father of genetics. Okay. So he introduced the concept genetics. So that is the reason we call him as the father of genetics. Okay. So, and he is not the scientist or the botanist. Due to his interest in the botany section or the plants, about the nature, so he started to work on some of the plants like a pea plant, otherwise the Pisum sativum. We call it as a Pisum sativum. The scientific name of a pea plant is a Pisum sativum. On that he started to work. Okay. So he tried he has taken a different characters, and he did a different experiment on that plant. Okay. So, and whatever he tried to do that, so he taken that in the form of a writing form. Okay. So then actually he is not as I told previously, he is not a scientist or the botanist. He is actually a monk in a church. Otherwise we also call him as a priest. So, and then whatever the record he has been taken, he submitted to the Natural History Society of Brunn in the year 1865. Okay. So at that time, the people don't have the knowledge about the genetics and whatever the concept about the genetics, how the evolution of a man takes place. They didn't have any concept about that. So that is the reason what happens, that theory got disproved. Whatever the Mendel has proposed, that theory got disproved. Okay. In the year 1900, so what happened, that theory has been republished. In the year of 1900, that theory has been republished by the scientists D. Rice, Correns and the Sedmark. Okay. These three scientists separately proposed the theory on the basis of a Mendelian concept. Mendelian concept only, they proposed the different types of theories. Okay. So, before we go deeper into this topic, the first I will tell that before the Mendelian concept, the people used to think that if a tall person marries a dwarf woman, okay, if the tall man marries a dwarf woman, whatever the offspring formation will take place, that will be the intermediate. This concept will be before the Mendel. So when Mendel started to work on that concept, so he disproved that whatever the previously it was that has been disproved by the Mendel, and he stated that if the tall person marries a dwarf person, either in those two, the one character is going to be seen in the offspring, like it might be a tall or it might be a dwarf, not the intermediate trait is going to be developed in the offspring. Okay. So then we are going to see, he worked completely by taking the pea plant. He completely worked on a pea plant. Okay. So there are some of the reasons why he taken the pea plant here means. So first the reason for taking the pea plant here, the first one, there is a number of varieties. There is so many number of varieties of a pea plant we are going to see. So that is one of the main reason why he has taken for the experiment. So first one is the number of varieties. Different types of varieties are present. So that was the main reason he has taken pea plant for the experiment. Okay. So next one, self-pollination. So before we are going to tell about understanding about the why he has taken, first we are going to understand that what exactly the meaning of a self-pollination. So in this case, what happens in the case of a self-pollination? So here commonly we are going to see the bisexual flowers. The same pea plant has a bisexual flower. In the bisexual flower, there will be a male part and a female part. Otherwise, we call that as an androecium and a gynoecium. Okay. So the androecium contains a stamen and the gynoecium contains a carpel or pistil, we are going to say. Okay. So in the case of a self-pollination, what happens? The transfer of a pollen grains, which is a male gamete, which will transfer to the female part of the same flower. We call it as a self-pollination. That means what happens? The transfer of the pollen grains from anther to the stigma of a same flower. We call that process as a self-pollination. So that is the reason he has taken for the experiment. They are a bisexual. That is the reason. For the continuation of a generation, they will undergo the self-pollination. So that will be helpful for the formation of our next generation. So that is the reason he has taken a pea plant. And the third condition he has taken here for the easy to cross-pollination. Cross-pollination here. So first we come to know about the self-pollination. Now we will understand about the cross-pollination. Cross-pollination means, in this case, what will happen? They will remove the male part of a flower and female part of another flower. Okay. So they will remove the male part of one flower and the female part of another flower. So they will cross-pollinate between those two different flowers. So then the formation of offspring will takes place. So that is what we are going to see. Okay. This is easy to cross-pollination. Okay. Otherwise we can say the transfer of a pollen grain from one flower to the another flower of a same species. We call that as a cross-pollination. Okay. Then the easy to culture. Easy to culture means, in this case what happens? The pea plants, you can grow them in an open field or in a small pot. Wherever you need, if you get a fertile soil, you can grow that in the open field or in the small pot also is capable of growing of those pea plants. Okay. And the last one here, the short period of a growth. Short period of growth, in a year we can get the lot of yield of the pea plant. Okay. Otherwise the Pisum sativa, we can get a large number of yield we are going to get in a shorter period. We can get 3 or more yields we are going to get in a short term. So these are all some of the reasons why why the Gregor John Mendel has been taken the pea plant as for the experiment. Okay. So first condition, the number of varieties, there is a different number of varieties we are going to see in this plant, and they are easy to self-pollinate, and they are also easy to cross-pollinate because of the two parts which are present in that flower. They are easy to self-pollinate, and by removing the one part from that each flower, we can make easy to cross-pollinate and easy to culture. We are going to grow that pea plant of the Pisum sativa in the open field, or in the case of the pot also we can grow. And in a shorter period, we are going to get the more number of yields we are going to get. Okay. So this is the reason why he has taken, Mendel has taken for the experiment. Okay. So next we are going to see, he concentrated, when he has taken the pea plant, he concentrated on the 7 traits or the 7 characters he has concentrated, and those are, first he concentrated on the seed shape, then seed color, then the flower color, pod shape, pod color, flower position, and the stem height. Okay. So each one we are going to see. This one we call that as a character. In that characters, whether it is a dominant trait and the recessive trait. Whatever the color we are going to see, whatever the character we are going to see, whether that character is a dominant or the recessive character, which character it is going to play. Okay. So in this case, the first one we are going to understand that the seed shape. In the case of a seed shape, there are two forms, otherwise two traits we are going to see. One will be the round, another one will be the wrinkled. Wrinkled means it has been, in Kannada we are going to say muduri irtakkantaddu anta heltivala. So that is what, okay. So in the seed shape, round and the wrinkled we are going to see. And when it comes to the seed color, what type of color that seed has? He concentrated on yellow and the green color. There are two traits we are going to see. One will be the yellow and another one will be the green. So when it comes to the seed shape, the round will act as a dominant character and the wrinkle will act as a recessive characters. Same in the case of a seed color, the yellow will act as a dominant character, the green will act as a recessive character. Then the flower color, either it may be a red or the violet and the white. So in this case, when we are going to see the flower color, okay, here we are going to see the red or violet color will act as a dominant and the white will act as a recessive. Color. So you already know the concept of a dominant and the recessive, which is going to be expressed in the next generation. We are going to call it as a dominant, okay. So which will not be expressed in the next generation, we will call it as the recessive, okay. So then the pod shape. In the pod shape, pod means where the seeds has been situated. That's what we call it as a pod, okay. Pod, P-O-D, pod, okay. Pod means where the seeds got situated, okay. You can see the pea seeds. If you see, they have been situated inside the pod. Inside the pod, peas has been situated. So that covering layer, whatever we are going to call, that is a pod shape. So either it might be a full, either it might be a constricted. Then the pod color, which type of color that pod has, okay. So either it might be a green or the yellow. So in these two cases, when it comes to the pod shape, the full will act as a dominant and the constricted will act as a recessive. The pod color, the green color will be dominant and the yellow will be the recessive, okay. Then the flower position. You might have seen in the plant, the flower will not be in the same position, right? It has a different position. Like if we consider here, the flower will originate from the axial bud, right? So here we call it as a axial, and this position we call it as a terminal, okay. So if the flower will originate from this position, we call it as the axial, and if the flower has been originated from the terminal region of a plant, okay. So that's what they have said, flower position. Either that flower present at the axial region of a plant, or it is present at the terminal, at the top region, what we are going to see, that it is present at the terminal region of a plant, okay. So here the axial will act as a dominant and the terminal will act as a recessive. And lastly, the stem height. Stem height, here we are going to see the tall and the dwarf. In this case, what happens? The stem height, either that stem is a tall, otherwise the complete plant is whether it is a tall or the dwarf. In the 10th standard, you might have, if you are a Kannada medium student, ಎತ್ತರದ ಸಸ್ಯಗಳು ಮತ್ತು ಕುಬ್ಜ ಸಸ್ಯಗಳು, okay. So in here we are going to see the tall will be act as a dominant and the dwarf will be act as a recessive. So these are all some of the characters which the Mendel got concentrated on these 7 characters, okay. So the seed shape, seed color, flower color, pod shape, pod color, flower position, then the stem height. These are the 7 characters what the Mendel or the father of genetics has been concentrated on that, okay. So next we are going to see So first one, here we are going to see the cross-pollination of pure line breeding. Cross-pollination of pure line breeding. The second one, hybridization. And the third one, here we are going to see self-pollination. Self-pollination of F1 hybrid. Self-pollination of F1 hybrid. So in this case, what he has done, first he has taken the pure line breeding. Pure line breeding, so whatever the genes which are homozygous, if we have taken, I will show you one of the example here. Just if we have taken height of a stem, right? Okay, we will go with the flower color. Flower color. Okay. So in the flower color, we are going to see two traits, that will be the red and white. Okay, the red and white are the two traits which are present in a flower color. Okay, so in this case, what happens? So we are concentrating on the first one, cross-pollination of a pure line breeding. Okay, so whatever the gametes here we are going to see, or the genotypes, whatever we are going to see, like for the red we are going to see the capital R, and for the white we are going to see small r. Okay, so these we call them as a pure line breeders, because that flower contains the homozygous genes. The whatever the, either it might be a red or either it might be a white. So those flower contain the homozygous genes. Okay, capital R, these are the homozygous genes, and the small r, these are also the homozygous genes. Okay, so he has taken those two pure line breeders and he allowed them for the cross-pollination. By the human involvement only, they will undergo with the cross-pollination. Okay, he allowed them for the cross-pollination. So this is the first condition. In the second condition, what will happen? The hybridization. In the case of hybridization, we are going to see here. So when the cross-pollination has been taken place, at that time, what happens? In the F1 generation, there is a formation of a capital R and a small r. So here it is a heterozygous red. So this process we call it as a hybridization. After the crossing of these two pure lines, right? Whatever the result will come, we call it as a hybridization. Otherwise, hybrid. We are going to, in the market, you might have seen that there are so many hybrid fruits and the hybrid vegetables you are going to see. That is all due to by the crossing of these pure lines, we are going to get the hybrids. So there is a word they use it, F1, right? F1 means filial one. Otherwise, we also call it as a offspring. F means filial. Filial is nothing but a offspring. Okay. So offspring. Okay. So this is what the hybridization process will take place. After that, he will take the two hybrid plants. Okay, two hybrid plants. He allows them to self-pollination. Self-pollination, you know, they are going to pollinate in between them only. Okay, there is no involvement of a human being will take place here. So they are undergoing with the self-pollination. After the self-pollination, whatever we are going to see in the result, we call that is the process what we are going to see in a self-pollination. Okay. So in the case of a self-pollination, the hybrids are going to be crossed. Okay. But in the case of a cross-pollination, the pure line breedings are going to be crossed. So this is how the Mendel has been conducted an experiment on the pea plant. Okay. So first one, the cross-pollination of a pure line breeding. The second one is a hybridization, and the third one is a self-pollination of a hybrid breeds. Okay. So this is how he has been conducted the experiment on the pea plant. Okay. So this is what we are conducted today's class, and tomorrow we are going to discuss about the further topic. Okay. Thank you, and if you have any doubt, please comment in the comment